it happening? Recording started. So, Michael, shall we start?
It's fine with me, thank you. Yes, we can start.
Yeah. So, good morning, everyone. I warmly welcome all the participants to this GYAN program on ribosomes flow model, theory and applications. It is a pleasure to have you all here. I would like to express my sincere gratitude to the Ministry of Education for their financial support in making this course possible. A special thanks to Professor Michael Marangliot for kindly accepting our invitation and offering this course. We are truly honored to have you here, sir. Lastly, I would like to thank IIT Roper for extending all the necessary support to successfully conduct this program. So now I would like to introduce our speaker, Professor Michael Marangliot. Professor Margliot is a leading researcher at Tel Aviv University with notable contribution in system biology, control theory, and neural networks. He co-authored several papers on the ribosome flow model and has extensively worked on contraction theory, switched systems, Boolean networks, and compound matrices. A recipient of the Dean's Award for Teaching Excellence and an incumbent of the System and Control Chair, he brings over 30 years of academic and research experience across engineering, biology, and computational modeling. After his postdoc, he served for 2 years in CUT's system as a scientific consultant and joined the School of Engineering, Electrical Engineering as a lecturer to become a full professor in 2014. Professor Margliot has courses on artificial neural network, computer architecture, pattern recognition, data structure and algorithms, control theory and system biology at Tel Aviv University for more than 20 years. So I request Professor Michael Marangliot to start the program with his first lecture. Over to you, Michael.
Thank you very much. Can you hear me?
Yes, we can hear you.
Excellent. And you can see the slides.
Yeah, we can see the slides.
Fantastic. Thank you so much, Arvind, for organizing this course. I know that, and for inviting me to give a lecture here. I know that this must have been a lot of hard work, and I appreciate it. And it's always nice to meet new people and to talk about my work and listen to good ideas. And I would like to encourage all the listeners to ask questions at any point. Just barge in and ask questions, because it really helps me to understand what is clear, what is less clear. Should I speed up? Should I slow down? Any feedback online is really highly appreciated. And if for some reason you can't hear me well or you can't see the slides, again, just interrupt and tell me, and I'll try to improve the situation. Okay. So the topic is the ribosome flow model, something I started to work about, I think, I started to work on, I think, about 14 years back. And today's lectures I will give using slides. Starting tomorrow, I will write on the board behind me. I will just write everything on the board behind me. I'm also making an effort to supply lecture notes to the people attending this course. I've sent Arvind already some notes for this first introductory lecture, and as the course goes along, I will send more and more lecture notes. I'm not sure if I'll finish everything on time. If not, I'll send Arvind all the lecture notes in a week or two, in the worst case. Then he will distribute it to all the course participants. Okay, so let's start talking about the ribosome flow model, theory and applications. Actually, so it's not exactly syllabus handed out, but as I said, I've sent lecture notes to Arvind and he will distribute them. Although we will speak about the RFM throughout, it is important to understand that there is a bigger perspective, a wider perspective, which is analyzing and manipulating biological systems and processes, which is a primary challenge for the 21st century science. And I would like to discuss how to do this efficiently using analytical tools from systems and control theory, which is my field of research. And I believe this is a very successful domain of study that still has far-reaching potential. So I will start speaking a little bit on this wider perspective of systems biology, but we will use throughout the RFM as a specific example in order to make things also more concrete. So the outline is what is systems biology. I will very briefly recall one definition by Sontag on this topic. And I will try to explain how systems biology can benefit from an engineering perspective and vice versa. How can the study of engineering systems, but also mathematics and physics, can benefit from modeling and analyzing biological systems. And as I said, we will speak throughout about the ribosome flow model. So one definition of systems biology by Eduardo Sontag, a field of study aiming to unravel and conceptualize the basic dynamic processes, feedback control loops, and signal processing mechanisms underlying life. So just from the words dynamic processes, feedback control loops, and signal processing, it is clear that mathematicians, physicists, and engineers may have a lot to contribute to this field. And there is a growing need for computational models in this field, and in particular in cell biology. This is because, first of all, new experimental devices produce more and more data, and computational models are needed to organize, integrate, and understand this data. And there are two fields where people actually manipulate the biological machinery. First is biotechnology, where we use living systems and organisms to develop or make products. For example, we can use the cells of some organism to produce insulin, human insulin, in order to provide this as a medicine for people who, for some reason, do not produce insulin properly in their bodies. And another related field is synthetic biology, where we use engineering principles to design and assemble new biological components. In both those fields, we manipulate the biological system, and then having a computational model can save us a lot of time in order to be able to test all kinds of hypotheses as to what will be the effect of our manipulations, to produce, using the model, falsifiable predictions, and in general, to do some preparation using the models instead of jumping directly to the lab. And this can save a lot of time and effort. So as I said, we will speak a lot about the ribosome flow model. This is the cornerstone of this course. Although I want to put it in the more general framework of systems biology. And the ribosome flow model is a model for something called translation, which is an important process in gene expression. So gene expression basically means that our cells constantly transform the information in our DNA into working proteins. And this process is called gene expression. It has several stages. Using the stage of translation, which is the stage we are interested in here, ribosomes flow along something called the messenger RNA or mRNA. read the information encoded on this mRNA and translate it into proteins. I will speak about this in a little more detail in a moment. And the ribosome flow model, or the RFM for short, is a model for the flow of ribosomes along the mRNA during translation. And I will describe applications, I will describe various properties of this mathematical model that can be derived using rigorous analysis, and then their biological implications, again, as a demonstration of how mathematical, physical, and engineering models in systems biology can be, I think, very useful. Any questions up to this point? Okay, as I said, please don't hesitate to ask anything. Actually, it makes me feel better to know that there is someone out there and not that I'm just speaking to the computer screen. Okay, so the central dogma of genetics, we have the DNA. I think everybody is familiar with this picture of the double helix here on the left. And this undergoes a process called transcription that generates something called the mRNA, and we are interested in the process of translation, where the mRNA is converted into protein. So think of the DNA as a recipe for producing a protein, and gene expression is all the process until this protein is actually produced. So here is a little bit more information. We have the DNA inside the nucleus of the cell, depending on what organism we're speaking about. But let's assume it is in the nucleus of the cell. It undergoes transcription, generating an mRNA. This leaves the nucleus, and then in the cell itself, it undergoes the process of translation. So the mRNA includes what we might think of as bits of information. They are called codons. Every codon includes 3 bits, and every bit can have one of 4 values. And this codon encodes for 1 amino acid. The protein is a chain of amino acids. So we have here a list of amino acids and complex machines called ribosomes. This is this big gray bulb here, attached to the mRNA and scan it codon by codon. Every time they read in a sequential manner a new codon, they wait for something called tRNA, transfer RNA, that carries the correct amino acid. When this arrives, it attaches to the ribosome and then delivers the amino acid, the correct amino acid, and slowly what happens is that we have a chain of generated amino acids, and as the ribosome reaches the end of the mRNA, which is marked by something called the stop codon, it detaches from the mRNA, releases this chain of amino acids, and this becomes, after a few more processes, the functioning protein. So what is important is, first of all, that the ribosomes must scan the mRNA in a sequential manner, codon after codon. They cannot bypass one another because the information is sequential. This is one thing. And another important property is that several ribosomes can scan the same mRNA in parallel. So at the same time, there can be many ribosomes of the same mRNA. And this form of pipelining allows the cell to produce more proteins for every time step. So as I said, the DNA code is made up of 4 letters. So this is a snapshot of a small part of this code, and each letter can be either A, C, G, or T. And it's interesting that this is a truly hybrid system in the sense that it is based on digital information, because these are just bits if you think of it. And this actually controls the continuous time world of chemical concentrations and the productions of proteins. So in some sense, it's like a software of a computer, a digital software of a computer that controls the continuous time process. So here is another schematic figure, and also some photo derived using some kind of microscope. So here what you see in blue is the mRNA with its two ends, 5' and 3'. 5' corresponds to the beginning of the mRNA. The ribosomes include two units, a large subunit and a small subunit. They attach together at near the start codon, which signifies where the ribosomes start reading the information. Then they scan the information codon by codon, and they generate this chain of amino acids, which becomes longer and longer as they traverse along the mRNA. And when they detach here from the stop codon, they release this completed chain of amino acids. And here you can see the same thing in the cell. So this is the mRNA. Those big bulbs are the ribosomes, and you can see the chains of amino acids as well. And these chains become longer and longer as the ribosomes approach the end of the chain. Okay, so how to model all this in a mathematical model? Why is it important? So let me start with some ideas on the modeling or describing the ribosome flow model, and then I will explain what is the importance of the analysis using the RFA. I should start by saying that the ribosome flow model is actually, it can be derived from something called the totally asymmetric simple exclusion process, which is a fundamental process in mechanical statistics, and a very important process. I wrote a paragraph or two about TASEP and also references. Sorry, the totally asymmetric simple exclusion process is also called TASEP for short. I wrote a few paragraphs on this in the lecture notes that I've sent to Arvind. By the way, would someone like that? Perhaps I can send a PDF also over this Teams system. Is there a way to send everybody the PDF file?
Yes, no problem. You please share, we can circulate it to everyone.
I'm sorry, Arvind, what did you say?
No problem, we will circulate it. You please send to me.
Okay, yes. I've sent it to you, I think, last evening.
Fine.
But if there's a problem, I can also distribute this using these team systems. I'm more familiar with Zoom, but I'm sure I will find a way as well. Okay.
Okay, fine.
Okay, so let's proceed. So as I said, TASEP is a very important model in statistical mechanics. In this model, we have a chain of sites, and particles hop from site to site in a unidirectional manner. And the hops are stochastic, so there is some stochastic rate of hopping. And a very important property is a property of simple exclusion, which means that a particle cannot hop into a full site. So a site can be either empty or include a single particle. So, and this corresponds to the fact that the ribosomes cannot overtake one another. So the ribosome flow model is a deterministic model that can be derived as a mean field approximation of TASEP. Assume that somehow the mRNA is divided into n sites. Each site can be a codon or a few consecutive codons, but in general we have n sites. Now we also have n state variables, xit, that take value, are normalized in 0, 1. and they represent, XIT represents the ribosome occupancy level at site I at time t. So if X3 is 0, this means that site number 3 is completely empty. And if X3 is 1, then this means that the site is completely full. The state space then is the n-dimensional unit cube. We have n state variables, and each one can take values in 0, 1. And there are n plus 1 parameters called lambda I, which are the transition rates from site to site. So let me describe now the equations of the RFM. Okay, so first of all, here is a picture. We have n sites. Each site is a codon or a set of codons, consecutive codons. And we have these state variables x1, x2, up to xn that tell us for each site how full it is. Again, 0 means that the site is completely free, and 1 that the site is completely full. And we have these lambda i's. Lambda 1 basically says this is a positive parameter that somehow tells us how easy it is to flow from site number 1 to site number 2. Lambda n minus 1 tells us how easy it is to flow from site n minus 1 to site n, and so on. Now here are the equations of the RFA. So we have n densities, x1 up to xn, and the equations tell us how these change in time. And in order to understand these equations, let's look at the equations for x2 dot. So this is the change in the density in site number 2. And this change in density includes two terms. This term, lambda 1 x1, 1 minus x2, is the flow from site number 1 to site number 2. And this term is the flow from site number 2 to site number 3. So this is just a balance equation that tells me that the change in the density in site number 2 is the density of particles entering from site number 1 to site number 2 minus the density of particles leaving site number 2 to site number 3. Now let's look at each term separately. So as I said, this represents the flow from site number 1 to site number 2. First of all, it is proportional to lambda 1, this positive constant that tells me how easy it is to flow from site number 1 to site number 2. Sorry. It is proportional to x1, which is the density in site number 1. So if there are more particles in site number 1, more particles flow into site number 2. And it is proportional to 1 minus x2. This is a very important term. This is the amount of free space in site number 2. So the idea is that as the density in site number 2 increases, less and less particles can enter site number 2. This is a way, this models actually the simple exclusion process in passive. As the site fills up, it tells the site before it, look, I'm filling up, I cannot receive any more particles. The particles, of course, are ribosomes, and the densities are densities of ribosomes, but the model is very general. You can also think about traffic, vehicular traffic along the road. Then you somehow divide the road into sections, say somehow in correspondence with the geometric properties of the road. And then the densities will be densities of vehicles along the road. You can also imagine some kind of evacuation corridor where humans leave some, I don't know, football stadium. And then the densities will be the densities of humans in each part of this evacuation corridor. So to summarize, this corresponds to unidirectional movement from site 1 to site 2 to site 3 and so on. And we have a soft model of simple exclusion because of this term 1 minus x2 that tells us that when the density in site 2 increases, the flow into site 2 decreases. And of course, this is the transition rate from site 2 to site number 3. Again, this is proportional to this positive constant lambda 2 that tells us how easy it is to move in general from site number 2 to site number 3. It is proportional to the density in site number 2 and to the free space in site number 3. Was there a question? Okay. The output rate from the last site is lambda n xn, and we give this a special notation, let's say R, and this is the rate of ribosomes leaving the mRNA, which is also the protein translation rate. So RT equal to lambda n xnt is the translation rate at time t. Now, a very important property of the ribosome flow model, or RFM, is that it is highly amenable to analysis. There's a question, please. Okay, it is highly amenable to analysis using various tools from systems and control theory, including contraction theory, monotone system theory, analytic theory of continued fractions, spectral analysis, convex optimization. Yes, please. If there's a question, please go ahead. Okay, I'll continue in the meantime. Convex optimization theory and random matrix theory. In this crash course on the RFM, we will discuss in detail some of these tools for analyzing the RFM, but I want to emphasize that some of these tools, especially contraction theory and monotone system theory, proved useful in analyzing many models from systems biology, not just the RFM. And today, in this introductory lecture, I will just give some introduction to what are these tools, what do they mean, how they can be used to analyze the RFM, and what are the biological implications of the analysis. So let me start with contraction theory, very general introduction to what this theory is. We consider an n-dimensional, an ODE here, x dot equals fx with n state variables. And assume that we fix two initial conditions in our state space, in our n-dimensional state space. One point is A and one point is B. And xta will be the solution at time t when we start at the time 0 in the initial condition A. And xtb, of course, is the solution at time t, when the initial condition at time 0 is B. Now we say that the system contracts if any two trajectories converge, approach one another at an exponential rate. What does this mean? There is some vector norm, so that when you look at the difference xta minus xtb and you apply this vector norm, it decays exponentially in time. Eta here is a positive constant, some positive constant with respect to the difference between the initial conditions. So as you can see here in the picture, the trajectories contract to each other at an exponential rate. Then we say that the system is contractive. And of course, it has to hold for some vector norm. What are the implications if the system is indeed contractive? First of all, imagine that there is a unique equilibrium. Imagine that there is an equilibrium, imagine that there is an equilibrium. So what is an equilibrium? An equilibrium is a point E, so that if you start at E, if your solution emanates from E, it remains in E for all time t. This is the definition of an equilibrium. Now, because any solution contracts to this solution, remember, contraction means that any two solutions contract, and xte is a solution. So any solution contracts to xte, so any solution contracts to E. So this means that all the trajectories converge to E at an exponential rate. And of course, E must be unique, because if there are two equilibrium points, then the solutions emanating from these two equilibrium points will not contract to each other. And this is a contradiction in a contraction system. Another important property is that, very important property, is that a contractive system entrains to periodic excitations. And I will speak about this in a moment. So let's apply this to the RFM. It turns out that the RFM is a almost contractive system. I don't want to get into the details today. It is almost a contractive system on our state space, which is the n-dimensional unit cube. And this means that there must exist a unique equilibrium point E, and all trajectories converge to E. So what does this mean in our model? First of all, it means, we go back to the equations of the RFM, it means that when there is some value E1, E2, E3, up to En, so that when I plug these values Ei in the Xi's, I get 0 in all the derivatives. This is the definition of an equilibrium point. At an equilibrium point, all the derivatives are 0. So what does this mean? It means that there are some steady state densities of each cell, and when you are in this steady state density, the flow into the cell and the flow out of the cell are equal. Because they are equal, the density remains constant. So this corresponds in some sense to a dynamic steady state. There is still a flow from side to side, the flow of ribosomes, but because the flow into the site and the flow out of the site is equal, the density remains constant. So what is the biological interpretation then? This equilibrium point will only depend on the lambda i's. It does not depend on the initial condition. So the parameters, the lambda i's, the transition rates, actually determine a unique steady state of ribosome distribution and also a unique steady state of protein production rate or synthesis rate. This is important because it says that when we want to analyze biological behavior, can say, okay, there is a relatively short transient. After this relatively short transient, we are already very close to the equilibrium or to the steady state because convergence is exponential, an exponential rate. And then to understand what's happening, we just need to understand what is happening at the equilibrium, at the steady state, after this short transient that only happens until we converge to the equilibrium. Questions?
Hello.
Yes, please.
Yeah, so this equilibrium state in biological terms, so can we say that the number of ribosome present on the DNA or on mRNA, so they are giving equal number of amino acids, so in equilibrium states.
So again, please, at the steady state, we have a constant number of ribosomes in each side, which corresponds also to a constant flow of ribosomes from side to side.
Okay.
And then you also have a constant flow of ribosomes from the last side. The constant flow, the flow from the last side is lambda n xn. At the steady state, it will be lambda n en. And this is the constant protein production rate. Lambda n en. Thank you for the question.
Yeah, okay.
Okay, so here is a picture of a simulation result. This is an RFM with 3 sites, just so that we can plot the trajectories. But when I speak about the analysis of the RFM, everything I say holds for n-dimensional RFMs for any n. And what you see here are trajectories emanating from 4 initial conditions that I mark now with my cursor. And all these trajectories in the x1, x2, x3 world converge to the unique equilibrium. So after a short time, we are very close to this steady state. Again, I think the interesting thing here is to, on the one hand, do the analysis, and on the second hand, try to understand what this analysis means from the biological perspective. And I always found this very interesting. Very interesting game to understand, first of all, what we can say about the model, and then what does it mean for the biology. Okay, as I said briefly, a contractive system entrains to a periodic excitation. I will speak more about this when I speak about contraction theory. But today I just want to explain what it means in the context of the RFM. Imagine now that these lambda i's that tell me how easy it is to move from side to side, they are not constants anymore. Now they are time-varying functions. So I think it's easy to think, if the RFM models traffic flow, then imagine that the traffic flow is controlled by periodic traffic lights. So these lambda i's, think of them as the traffic lights between one section of the road to the next section. And imagine they all vary, they're all positive, but they are now positive functions with a joint period capital T. And then we can prove, using contraction theory, that all the densities in the ribosome, in the RFM, will converge to a T periodic pattern. So when you look at H density, it will also change periodically with the same period as the traffic lights. And also, of course, the protein production rate will also change in a periodic manner. So I think it's easy to think about this in terms of something called the green wave, when we operate traffic lights in a jointly periodic manner along the traffic road. Sometimes we would like the traffic also to oscillate in a periodic manner, just like the traffic lights. And then this is called the green wave phenomenon. Physicists are very interested in this. In the context of translation, there are many periodic processes in the cell. For example, there is a process called the cell cycle, where every cycle generates two new cycles. Every cell, generates two new cells. And this happens in a periodic pattern. And then we would like somehow that all the processes inside the cell during the cell cycle will also entrain to this periodic pattern. So in the context of the RFM, what this means is that if you somehow vary the lambda i's in a periodic pattern, you are guaranteed a capital T periodic pattern, or a periodic pattern with period capital T, you are guaranteed that the ribosome densities and the protein production rate will also converge to a periodic pattern of the same period, capital T. Let me first of all show you a figure demonstrating this, and then we'll go back one slide. So imagine again, just for simplicity, that we have three sides. And now instead of constant rates, we take the rates here. So these rates are all positive, and they are periodic with period capital T equal to 1. If you add 1 to this T here, you will get 2 pi T plus 2 pi, and because sinus is 2 pi periodic, this is just sinus 2 pi T. This is certainly periodic because a constant function is also periodic. And this is also periodic with period capital T equal to 1, and so on. So is this one. You see they have different phases, but it doesn't matter. All that matters is that there will be positive functions and share a common period, in this case capital T. And then when we simulate the RFM with these rates, starting on the arbitrary initial condition where all the initial densities are 1/2, every density converges to a periodic pattern. And if you check the period here, this is something like between 230 and 2.5, 3.5, the peaks. So also period capital T equal to as predicted by the theory. So just going back, the RFM is almost contractive on our state space, which means that if we use periodic initiation and or transition rates with a common period capital T, the trajectories will entrain to this periodicity. Of course, in the RFM, it means that the ribosome distributions and also the synthesis rate converge to a periodic pattern. And I would just add that this property called entrainment, so if you think about this periodic excitation, periodic input as some kind of T periodic excitation, and then the system entrains to this periodic excitation in the sense that the solutions also converge to a periodic pattern, this property called entrainment, and it is very important. So in both natural and artificial systems, for example, many processes in our cell converge to the period of 24 hours, the period of the solar day, because we are all the time exposed to our eyes to this 24-hour solar day. And if you look, for example, if you consider the secretion of hormones in our body, you will see that many of these have a 24-hour period. In artificial systems, for example, when we connect a synchronous generator to the grid, let's say that the grid has a frequency of 50 Hertz, it's very important that the generator will synchronize with the grid and also work in a period of 50 Hertz. So this entrainment is really a very, very important property. And in contractive systems, you can easily prove that they indeed entrain. More questions, please?
Yeah, I have a question.
Yes, please.
These periodic functions must be a continuous functions, right?
Yes. Yes.
Okay.
Yes. I didn't say that, but when we study contractive systems, we had this model x dot equals fx. We usually assume that f is c1, so it is the vector field is continuously differentiable vector field. We need this for the theory of contractive systems. Yes, that's a good question because it might be interesting.
Yeah.
Some cases, like traffic lights, perhaps, where there is a jump.
Yes.
Yes. More questions, please?
Yeah, I have one question. How do you ensure like these models will give the better predictability over the others? How we are going to ensure the predictability of the model?
Well, you can, and we did that together with a computational biologist, Amir Tuller, who I've been working with for many years. He collected a lot of information about translation, and also there are big databases where you can take information about translation, and then you can try to fit the model behavior to the data. That's an interesting problem because you have to identify the appropriate lambda i's.
Okay.
An interesting question is how to do this efficiently. The model is, of course, non-linear. So
Yeah, I understood that one. Once we fit the model, but in the future like future predictability of the model, for example, if in a given conditions after fitting the model behaves properly, but how you are going to use this model for the future prediction, and how you are going to ensure that whether it will give a correct results.
So, for example, let's speak a little bit about an evacuation corridor for a moment. I think it's a little easier to speak about this. So imagine that you have an evacuation corridor and there are turns along this evacuation corridor, and when there is a turn, so you will divide it into cells according to the geometry, and then maybe you will put a camera in each section of this evacuation corridor, and you will measure the amount of humans there. And now you have, say, some continuous data from these cameras on how the people flow from one cell to the other, and you know the densities, you can measure the densities in each cell at each time. And now we will try to fit an RFM with proper lambda eyes that has more or less the same behavior. And now once you did that, then you can say, for example, imagine that you increase one of the corridors somehow, or you change something in one of the corridors, and you know, or you can estimate how this will change lambda I. Lambda I is how easy it is to move from one corridor to another. So maybe you can say, okay, if I rebuild this corridor a little wider, then lambda I will increase by 10%, and then I can plug in this increase in 10% in my RFM, and try to predict what will happen when I increase lambda I by 10%, and give this as a prediction to the engineers who want to redesign this evacuation corridor. Now, in the context of translation, the lambda eyes correspond perhaps, as I said, when the ribosomes move from side to side, they have to wait for something called tRNA that brings the correct amino acid. And then this waiting time is somehow proportional to the lambda I, or to 1 over lambda I. If you wait longer for the tRNA, if you have to wait longer, if these tRNAs are not so abundant, then this means that your lambda I will be smaller, because the rate of transition will be slower, because you have to wait a long time on average for the tRNA. So there are many open problems still on how to fit the parameters, how to make a good identification of the parameters, and how to use it for prediction. All these are open questions, and I think they are interesting research questions, and I think it's good if this crash course will also lead to some research work on these questions. Many things I don't know the answer, but I think it can be an interesting research direction.
Thank you very much. I understand the concept of. So based upon the lambdas, we are going to use the how it is changing in the future, like but it will not guarantee the predictability, but it will give the direction of the predictability.
Yes.
Thank you.
Yes. Thank you. Okay, so let's talk a little more about another direction that proved useful in the analysis of the RFM. And this is the analytic theory of continued fractions and spectral analysis. And I will speak more about this during this course. Today, I'm just giving some intuition. So, again, just for simplicity, consider the case n equals 3. And what happens at the equilibrium point? We already know that there exists a unique equilibrium point in our state space, which is a 3-dimensional unit cube. So when we plug in here, instead of xi, we plug ei, then all the derivatives are 0. So we get lambda 0 multiplied by 1 minus e1 is equal to lambda 1 e1, 1 minus e2. This is the equation we have here. This just says that at the equilibrium, the entry rate into the first site and the exit rate from the first site to site number 2 are equal. And then we also have these equations that tell us that all the other entry and output rates from each cell to the other are all equal. So we get this chain of equalities that define the equilibrium state. And of course, the solution only depends on the lambda i's. Now we can do some manipulation with these equations. First of all, from these two equations, we can represent E2 as a function of E3. You can see it easily from these two equations. By the way, I'll also be happy to send Arvin these slides. I'll do that after we finish today at 10 o'clock, my time. And then from this equation and this equation, we can express E1 as a function of E3 and E2, and we already have an equation for E2, and when we plug it in here, we get an equation for E1 in a form that is called a continued fraction of E3. Okay, you see why this is called a continued fraction. We have E3, and then division, and then E3 again. And in general, every EI can be expressed as a continued fraction of the last density E3, or in general En. Now there is a close connection between continued fractions and spectral analysis. Spectral analysis is the theory of the eigenvalues and eigenvectors of a matrix. So first of all, again, if we go back to the continued fraction, let R denote the steady state production rate. R of t is the steady state production rate, and R is the steady state. So R of t is the production rate at time t. Let us denote by R steady state production rate. So this is after we Xn converge to En. Then R will depend on all the lambda i's, and it turns out that we can also write it in this elegant continued fraction form. This follows immediately from what I showed you in the last slide, because remember, we expressed everything using En, and En is just R up to multiplication by lambda n. So we have this continued fraction expression for steady state production rate. And continued fractions are closely related to the spectral properties, the eigenvalues and eigenvectors of tridiagonal matrices. And this can be used to provide a spectral representation from the lambda i's to the steady state production rate. So let me just show you the spectral representation. We will speak more about this in the following lectures. The idea is the following. You have the RFM. The RFM includes n+1 parameters, lambda 0 up to lambda n. Take all these parameters, you put them in an n+2 by n+2 symmetric, non-negative, irreducible, tridiagonal matrix, which is the matrix A given here. So you put all the lambda i's. First of all, you take them to the power of -12. They are positive numbers, so this is well defined. You put all the lambda i's here on the superdiagonal in this matrix and on the subdiagonal, and all the other entries are 0. So this is a symmetric, non-negative, irreducible tridiagonal matrix. Now, in such a matrix, all the eigenvalues will be real, and they will all be different. So we can order them with alpha n+2, the maximal eigenvalue. And then it turns out that the steady state production rate in the RFM satisfies this equation here. The maximal eigenvalue alpha n+2 is exactly equal to R to the power of -12. So what this means is that I don't have to simulate the RFM, wait till convergence, and then measure R. I can take the lambda i's that define my RFM, put them in this matrix, calculate the largest eigenvalue, maximal eigenvalue, alpha n+2. Then this equation will give me the steady state production rate in the RFM with these lambda i's. So it's a nice way to represent the steady state production rate. And this gives you a spectral representation of the mapping from the lambda i's to R. What does it mean, a spectral representation? It means that it depends on eigenvalues, or in this case, the maximal eigenvalue of some matrix. And the spectral representation has many interesting implications. First of all, it says that we can calculate R numerically very efficiently using algorithms for computing the maximal eigenvalue of a tridiagonal matrix. This can be done very efficiently in a robust manner with small errors, small numerical errors, even when n is very large. Another interesting application is the following. We can prove now that the function R is a strictly concave function of the lambda i's. So the steady state production rate is strictly concave function of the lambda i's. If we make a plot here are the lambda i's. Here is R is a function of the lambda i's. It will always have this shape of an upside down bowl of food. And this is important when we try to ask, how can we maximize R? How can we find lambda i's under some constraints that will maximize my steady state production rate? Okay. perhaps I will stop here. We have 5 more minutes, but I don't want to. We have more time today. We have more lectures today, so we will continue in our next lecture. And also, I want to give 5 minutes. If anybody has questions, that's maybe a good time to ask now.
Excuse me, sir.
Please.
Sir, do these models follow some principles of fluid mechanics also?
I'm sorry, but somehow I just a minute. Somehow I can't hear you. Just a minute. I don't know if it is something I pressed or, but somehow I can't hear you. Is this Monique?
Yes, sir. I wanted to
Yes.
ask that do these models use some principles of fluid mechanics also?
Well, they can be derived from PDEs that describe fluid mechanics. I can give you a reference about this if you send me an email. And also, they can be derived from TASEP, which I think can also be, is one model for flow among a large set of models that also include models from fluid mechanics. There is a nice paper, a survey paper by several physicists on TASEP and its solution using matrix products. And in this paper, they speak about several models of PDEs for the flow of particles, maybe with some kind of exclusion. And then they put these models in a wider perspective, which also includes PDEs.
Okay, sir. Thank you, sir.
Hello, Michael. Apart from the blocking mechanisms, like due to the volume exclusion principle that two ribosomes cannot overtake each other, is there any blocking mechanisms that has been seen in this translation or transcription process?
Well, first of all, it's very nice to meet you again, Aditi.
Yeah.
We once in Germany, where
Yeah.
you postdoc. I still remember the very good restaurant that you took us all.
Yeah.
It's
Thanks.
nice.
Yeah.
And, okay, so that's a very good question. Let me say something that I think I forgot to say. Can you still see the slides?
Can I see the? Yeah. Okay.
So about blocking, I forgot to say something important, and Aditi's question reminded me. When we look at these equations, maybe I said it, I don't remember. When imagine for a moment that x3 increases, so the density inside number 3 increases. I think we have to finish, but imagine that the density inside number 3 increases, then this term, so x3 goes to 1, this term will go to 0, and then x2 dot will only have this positive term. So when x3 goes to 1, x3 fills up, x2 dot is positive, so x2 also starts to fill up. And then when x2 fills up, x1 will fill up. So what happens here is that the RFM can actually be used to model and analyze the evolution of traffic jams, just like TASEP. And the traffic jam will evolve in a direction opposite to the direction of flow. If the flow is from site number 1 to 2 to 3, the traffic jam actually evolves from 3 to 2 to 1. So I just wanted to say that, and I don't want to continue before longer than 10. So Aditi, if you send me your question by email,
Yeah.
I will answer by email
Sure.
the moment I get you.
Sure.
Thank you very much, and it's nice to see that you are participating today.
Yeah. Yeah. Oh, thank you.
Thank you, Aditi. Thank you for reminding me this topic. So I suggest that we stop now, and if that's okay with you, Arvind.
Yes, we can stop now. We'll meet at 2 o'clock Indian time for the next lecture.
Thank you very much. I'll be here.
Thank you all. So we'll close this session for now, and then we will meet at 2 o'clock.
Thank you very much.
Thank you.
If anybody has questions, please send me an email.
Thank you.
Send me directly. Thank you.
इसको क्लोज करो, इसको close कर दो।
एक एक